Hi, Dr. Mella. This is my final Ereport for cell and Molec. This is about the story of a cell, and it's pretty much like the reality show of the cell as told by membrane proteins. It's about how the cell interacts and communicates with itself and the environment around it. The cell membrane is what really interacts with the environment. The cell membrane is a lipid bilayer, and the lipids have a hydrophobic tail and a hydrophilic head. Since the hydrophilic heads have no problem with water, they face outward, and the hydrophobic tails huddled together. This creates a miscible structure. But membrane lipids in us don't just have one hydrophobic tail, we have two, and most of the time there's a kink due to a double bond. This means that the membrane lipids can't make a miscible structure. The hydrophobic tails still pack together away from the water, but they do it tail to tail like this. They also can't have edges exposed to the water, so they create a hollow ball called a liposome. This need for the hydrophobic tails to be away from the water is what causes the membrane to self-seal, and it's what makes it such a strong barrier. The lipids can't just pop out and move around once they're sealed. Instead, they use lateral diffusion to move around. They also undergo transverse diffusion, but the membrane is asymmetrical, so the function of the phospholipids aren't the same on one side as they are on the other. So there is an enzyme called flippase, which moves them back whenever they do transverse diffusion. Lipids are a great barrier, but they don't give the cell function. There are membrane proteins that make up about 50% of the cell membrane, and their main function is transportation. There are different ways for molecules to be moved in and out of the cell. There's active transport and there's passive transport. In passive transport, small non-polar molecules can easily diffuse across the cell membrane. Uncharged polar molecules can also diffuse across the cell membrane as long as they're small. The larger they are, the longer it'll take, and this is shown on the left. On the right is for anything that's charged or ions, because they're blocked from diffusing across the membrane on their own. They require facilitated diffusion from channels or other passive transport membrane proteins. Neither of these require energy because they're moving things from a higher concentration to a lower concentration, so it's what the molecules already want to do. Active transport, on the other hand, moves things from a lower concentration to a higher concentration, and this always requires energy. That energy can be from ATP, light, or coupled transporters, where the passive transport powers the active transport. An example of active transport is bacterial cells that use bacteriodopsin as a proton pump. Bacteriodopsin is a membrane protein that has a retinal in it. Whenever the retinal is hit with a photon of light, it changes shape. Whenever the retinal changes shape, it creates a change of shape in the bacteriodopsin. And with that change of shape, a proton is forced from inside of the cell to outside of the cell. This creates a neutral pH inside of the cell so the cell can live, and a high pH outside of the cell. Active and passive transport can also work together, like in the sodium-potassium pump. The sodium-potassium pump maintains osmotic balance in our cells by pumping sodium out of the cell to prevent water from coming in. In image 1, there's sodium outside of the cell, and it wants to go in, but it can't because there's a gate on its channel. There's potassium inside of the cell, which can go out, but it doesn't want to because of the negative charge in the cell it's content. This is creating not only a concentration gradient, but a membrane potential. In the second image, a signal comes in and opens the sodium pumps, and that makes the sodium rush in, forcing the potassium out. This continues until the sodium and the potassium reach an equilibrium. In image 3, the signal has left from the sodium channels and they've closed. Now, the sodium-potassium pump works to pump all of the sodium back out and to replace the potassium inside of the cell, and it uses ATP as its energy source. Membrane-bound transport proteins don't just form in the membrane. They are made within our cell and they travel along the endomembrane system to get to the places they need to be. The endomembrane system consists of the nucleus, the ER, the Golgi, endosomes, and lysosomes. The endomembrane system has a secretory pathway and endocytic pathway. The secretory pathway can secrete things from inside of the cell to the outside of the cell, while the endocytic pathway can take things from outside of the cell to inside of the cell. To get proteins into the nucleus, they must enter through the nuclear pore. So the nucleus has two membranes, an inner membrane which is connected to the nuclear cortex and the chromosomes, and an outer membrane, which resembles the ER. The reason everything has to go through the nuclear pore is because if it were to fuse, like with a vesicle, then it would be trapped in between the two membranes. To go through the pore, the membranes have to have a nuclear localization sequence, which the nuclear transport receptors will recognize. The receptors then connect with the pore fibrils and deliver what they're carrying into the nucleus. Unlike the nucleus, proteins on a vesicle can fuse with the ER, or they can be made into the ER. A ribosome begins to translate an mRNA into a protein, and as it's doing this, an RNA signaling sequence is translated and the amino acid is taken to the ER. The ribosome is bound to the ER membrane and continues to translate the mRNA. While the protein is translated, there's a stop transfer sequence. This is a hydrophobic region, so it'll be embedded in the membrane. And then there can be a start sequence, which is a hydrophilic region, and it's started back out through the ER. Each time it goes from a stop to a start sequence, there's a new loop, as you can see in the bottom. There's multipass proteins, so each one of these has a new start and a stop sequence for every turn. Once the proteins are embedded in the ER, they can move to organelles by vesicles, but they can no longer leave the membrane. The vesicles start with cargo receptors that are transmembrane proteins. On the inside of the cell, the cargo receptor binds to the cargo, which is the protein. Whenever this happens, there's a change in the structure of the part that's on the other side of the cell membrane. This change is like a flag for adaptin to come gather it together. While the adaptin is collecting the cargo receptors, it's also grabbing Rabs and V snares. Clathrin weaves around the adaptin and it pinches the vesicle, causing a clathrin pit. The pit tightens more and more, and then dynamin comes in and pinches the vesicle off the membrane. The vesicle is taken by walking proteins, and the clathrin pit releases. As the vesicle moves along this, the Rab is interacting with tethering factors from organelles around it. Whenever the Rab interacts with the correct tethering factor for where it wants to go, it binds, and then the T snares from the acceptor and the V snares from the vacuole interact and they twist. This twisting brings the vacuole closer to the membrane and they can fuse. They don't always fuse right away. Sometimes it's delayed and the vacuole will just sit there until the acceptor is ready for whatever it's carrying. Vesicles can have two directions. They can come into the cell or they can leave the cell. Coming into the cell, they start by fusing with the outer membrane, and then they are taken to endosomes. Endosomes take the cargo off the receptors and then split into two. So there's one endosome with the receptors that goes back to the outer membrane, and then there's another endosome with the cargo. The half with the cargo then goes to a lysosome for cellular digestion. Going out of the cell, they start at the ER. The cargo receptors are membrane-bound proteins that bind to cargo or proteins in the ER. When they bind to the proteins in the ER, the receptor on the other side changes its shape. This change in shape is what adaptin uses to gather the signaling receptors. At the same time, adaptin is gathering Rab proteins and V snares to be a part of the vesicle. Once the adaptin have gathered everything, clathrin starts to weave around them and create a pit. This clathrin pit tightens until dynamin comes in and forms a ring around it to pinch it off from the membrane. This is what you're seeing at the top picture. Once the vesicle is free, it's carried on microtubules by a motor protein. The walking protein moves the vesicles until the Rab protein interacts with the right tethering protein. Rab proteins are like a membrane identifier. They tell the tethering proteins where they are coming from, and that's how the tethering proteins decide if they should combine or not. If the tethering protein and the Rab protein combine, the next two things to combine are the T snares and the V snares. The T snares are on the acceptor membrane, and the V snares are on the vesicle. When the T snares and the V snares combine, they twist, thus pulling the vesicle closer to the membrane. Eventually, they're so close that they fuse. Sometimes fusion is delayed, so the vesicle is just docked and held in place until the right signal is given to complete the fusion. Normally, the vesicles go from the ER to the Golgi, and then from the Golgi they go to the outer membrane. They are modified in the Golgi, which gives them function in the outer membrane. One of these functions is a G-protein coupled receptor. G-protein coupled receptors are surface receptors. So GPCR is a receptor in the membrane, and when there's a signal, the shape changes at one end, and that shape change makes its way all the way to the other end. At the other end, there are three subunits, alpha, beta, and gamma, that make up the G protein. The alpha subunit is bound to GDP when it's inactive, but the shape change makes the alpha subunit let go of the GDP and bind to GTP. This is activated, and the alpha subunit can activate other processes and signal second messengers. In this picture, it's the alpha subunit has activated phospholigase C, or PLC, which cuts lipids. It cuts a PIP2 lipid in half. The head is now called IP3, and the tail is DAG. IP3 serves as a second messenger and opens a calcium channel in the ER, and DAG stays in the membrane and activates protein kinase C. Protein kinase C phosphorylates channels to allow more calcium in through the outer membrane, and this creates a signal boost and activates commodulin. This activates another kinase and creates a cellular response. So the one signal that came from the G protein linked receptor went to the G protein, and then that activated phospholigase C, which activated IP3 and DAG, which opened the calcium channels, which created a cellular response by binding with commodulin. So far, this is a lot about how the environment interacts with the cell and what goes on the inside and the membrane proteins. But now we're going to look at it as how the cell interacts with the environment. To look at that, the structure of the cell is important. The structure is called the cytoskeleton. The cytoskeleton is an intricate network of proteins and filaments that go through the cytoplasm. It's built with three different types of protein filaments: intermediate filaments, microtubules, and actin filaments. And each of these filaments give a different function to the cell. Intermediate filaments give the cell tensile strength. That is, when there's pressure on the cell, it can distribute the pressure through the intermediate filaments. This is because of how they're made. They're made like ropes. Intermediate filaments are made of strands wrapped together to make dimers, which are wrapped together again and again to make a rope-like structure. This allows them to stretch and pull, just like bridge cables are made. There are 4 classes of intermediate filaments, and depending on the type of cell, they might have more of one than another. Keratin filaments are found in epithelial cells. Vimentin filaments are in muscle and connective tissues. Neurofilaments are obviously in the neurosystem and neurons. And nuclear filaments are found in the nuclear envelope. This is a picture of the intermediate filaments in a cell. And you can see that they're everywhere throughout the cytoplasm. Next, there are microtubules. Microtubules are found in most cells. They're long, stiff, hollow tubes made of tubulin. They are made of alpha and beta subunits that alternate. So they started as centriole and are made from there, but they don't last long. They're more functional than structural. They also have a function in mitosis. The microtubules are used in anaphase to separate the sister chromatids and move to opposite sides of the cells. The stability of microtubules depends on what it's bound to. When they're first made, they're connected to GTP, which keeps them stable. Over time, this is cleaved to become GDP, which is less stable. When there's GDP at the top, it'll all just fall apart unless it's anchored to somewhere else, giving it stability. One of the functions of the microtubules is it allows motor proteins to walk along them. They were mentioned earlier whenever we were talking about vesicles. Different motor proteins walk in different directions, and they're fast. And in an article I read recently, their speed was measured to be half the speed of sound. Actin filaments are the final cytoskeletal fibers. Actin filaments are thin and flexible, and they grow similarly to microtubules, but they don't have an origin. They are commonly in the cortex underneath the cell membrane, but one of their biggest functions is cell crawling. Cell crawling is a 3-step process. First, the cell pushes out protrusions, and then the protrusions adhere to an external surface, and then the rest of the cell follows. In the first step, a sheet of Lamopodia is pushed out, which has a meshwork of actin, and then a smaller protrusion, called Filopodia, is pushed out, and they only have a small bundle of actin. These structures make contact with the external surface, and they use integrins to sense if they can crawl on it. If it's too slippery, then they won't attach, and if there's nothing there, then they won't attach. The integrins are connected to the actin in the cell. When the Lamopodia attaches to something, the actin must pull itself forward, and this is the flowing part of the cell. It's literally rolling over where it's attached. Not all cells crawl. Most cells stay in place, but that wouldn't mean much if every cell just stayed in place. They need to be connected to each other. There are 5 major types of attachment. First is a tight junction. Tight junctions seal cells together, and nothing can get by. They do this by making a zipper with proteins called claudulins and oculins. There are mechanical junctions, which consist of adherins, desmosomes, and hemidesmosomes. Adherins and desmosomes connect cells through cadherin. They not only link the plasma membranes together, but they also link the actin inside of the cells together. This allows for mechanical stress to not only be distributed in the cell, but also to surrounding cells. That's why whenever you push on your skin, it doesn't just rip. It's because the pressure is being distributed throughout your whole skin. Hemidesmosomes are a specific type of this junction. They bind epithelial cells to the basal lamina, but they work in the same way as adherins and desmosomes, which is good. If we didn't have those, our skin would be slipping everywhere, and that's just kind of gross. The last type of junction is a gap junction. Gap junctions are very private. They're used for 2 cells to communicate. Structurally, they aren't that functional, but they can relay signal very quickly. There are small connexons that are like pores connecting one cell to another, and they can be shut or they can be opened, and they send signals along. It's like playing a game of telephone. This is the life of the cell, but the boring, everyday cell stuff. Proteins are embedded in the membrane in the ER, and they can be shuttled from organelle to organelle by vesicles. When the proteins are in places they need to be, they have functions like transporting molecules across a membrane or being receptors. Looking at the cell as a whole, it's largely concerned with the cytoskeleton. The cytoskeleton is made of different filaments, and those filaments help with different functions, like cell crawling and cell junctions. And overall, all of those things are what makes our cells functional, and this has been my final cell molecular report. Thanks.